Good evening. My name is Kent Dix, and I'm CEO of Life 365. And welcome to the second session of the new normal. This is our first episode titled COVID-19 Testing, Variants, and Vaccines, Our Future Ahead. Before I introduce our panelists for this evening, I want to take a minute and thank all the healthcare providers, first responders, school teachers, family caregivers, and many more. Thank you for all your service and support. I also want to let you know that everyone will be on mute and that we will accept questions via chat, and we'll try to entertain a few questions throughout the webinar. We have reserved some time for questions at the end. If we don't get to your question, we will answer them after the webinar and post the recorded video on life365.health website or www.newnormal.buzz tomorrow. Now, I want to take a minute and introduce our distinguished panelists and have them provide a quick 30-second introduction. Kathy Petty, I guess I'm going to do the introduction for you. Kathy Petty is CEO of Modex and HealthSpring Global. Dr. Petty is a health technology executive and in vitro diagnostic expert with a unique combination of operational, clinical, and entrepreneurial expertise focusing on molecular diagnostics, genomics, consumer health, and artificial intelligence. Dr. Petty is founder and CEO of Modex AI, a knowledge domain company integrating her passions for diagnostics and data science. Dr. Petty was previously Chief Medical Officer of Ancestry.com. In addition, we're proud to have Tom Leggett, Director of Business Development for Sonora Quest Laboratories. Tom has over 25 years in healthcare, spanning emergency paramedicine, clinical research, and laboratory business development. In his current role as Director of Business Development for Sonora Quest Labs, he identified evolving needs in healthcare industry as they pertain to diagnostics and immediately adjacent fields. He then led a team to develop solutions for those needs through either internal innovation or partnerships with outside entities. Okay, let's get into it. When we ended season 1 of the new normal back in June 2020, we had 5.7 million people in the world that had been diagnosed with the coronavirus. Today, there have been over 113 million cases. The US had 1.7 million that tested positive, and today we are at 28.4 million. The US had 120 deaths at the time from coronavirus, and today we've had over 500,000. Last year at that time, the panel estimated that we would hit 200,000 deaths by September, October, which we exceeded, unfortunately, and 1 million deaths by spring, summer of 2021. Kathy, are we still headed to that trajectory, especially with the variants that are now showing up?
Those statistics are absolutely devastating. And I think everyone on this call has experienced it personally or have had family members and friends who have suffered from COVID-19 and hopefully seen through the end of it. But many of us have also known close family friends who have perished. I think at this point, though, we should really think about our optimism for the future. I am optimistic that it will get better. I am optimistic that the vaccines are going to make a significant impact on bringing us back to our normal, which is going to be a new normal, not necessarily never wearing masks again. But I think seeing the tremendous strides that our vaccine manufacturers have made have given me great hope that 2021, we're going to see some really good changes.
I'm going to stay with you just for a second. So what do you think that new normal looks like, right, in the future? I know I've heard it said that we're probably going to be wearing masks for at least another year, potentially. We may have to get vaccines every year, right, from that as well. Do you think that, I'll just say it openly, do you think that we'll have another session like we're going through right now as far as the COVID-19 in our life, or are viruses becoming more prevalent in our society where we'll see them again in our lifetime like this?
Yeah, I always like to think from a perspective of context. And what we all know, what we've all lived with in our lives right now is influenza. Influenza is something that we've lived with since we've been born. Influenza is a little different than COVID. You're like, well, how is it different? Well, first, I'd like to say influenza has a different rate of infection. And when you look at COVID, it might be one person infects 4 people, whereas if you look at influenza, one person infects maybe one to 2 people. So the infection rate is different. The vaccination success, however, so far looks like COVID will be the winner, where you will be more protected against COVID based on these new technologies with vaccination than with influenza. So I think we should think about, we will live with COVID. COVID is going to be something very similar to living with influenza, with obviously much more disease severity associated with COVID. More deaths are associated with COVID, more hospitalizations, and a lot more long-term comorbidities from having COVID infection. So at the end of the day, we've learned how to live with influenza, and we are going to learn how to live with COVID. Influenza, we are vaccinated seasonally. I suspect that we will have a similar trajectory for COVID. Influenza, we did not routinely wear masks, although in Southeast Asian countries, even during influenza season, it was part of the culture to wear masks. So I have a feeling mask wearing is definitely going to be in our future for at least the next 12 to 18 months.
Right. All right. Makes sense to me. So Tom, the question I had for you is more from, the testing perspective. We started testing back in the March timeframe, and that was more of a PCR test, right? That was started,
Right.
and then rapid tests started coming out. I'm still a little confused about, antibody tests and, PCR tests and other tests that are kind of out there and how they kind of work together. Do you do an antibody test first, or do you do a PCR test? And, how do they kind of relate together, and how have they evolved, over the last probably year or so to, well, let's just say, how do they evolve over the last year or so, but how do they also relate to the vaccine as well, testing and the vaccine?
Sure. Well, Kent, you're not alone in your confusion. I can
Okay.
tell you, that. So really, there are the three main tests that are out there, and each one of them serves a role in the pandemic. And the first one is that RT-PCR diagnostic test. It's looking for the viral load or looking for the active infection in those patients, and that's your nasal swab. It's really considered to be the gold standard out there right now because it has the best performance. In other words, it has the lowest limit of detection. It can really find minute traces of virus in your blood, or excuse me, not in your blood, but in the nasal specimen. It does have some drawbacks though, in that it has to go into a central laboratory to be tested. So right now at Sonora Quest, we have a little bit less than a 24-hour turnaround time on that, which is pretty good. I think it's very acceptable.
I mean, at the beginning, it wasn't Sonora Quest I'm complaining about, but at the beginning, they were saying it was taking 7 or 8 days sometimes to get a test from a PCR test.
It actually even went out beyond that.
Yeah.
But it was for pretty much all the labs out there, including Sonora
Yeah.
Quest. And we had a global shortage of equipment, a global shortage of consumables and reagents, and I mean everything was hard to find, right? So that was a lot of the problem right there. I think back at that point, Sonora Quest was able to do about 6,000 to 7,000 PCR tests a night. We're able to do 35,000 to 40,000 PCR tests a night right now. We're only really doing about 10,000, so we're way under capacity, which is a good thing. But that's part of the reason why we're knocking it out of the park with a really quick turnaround time on that.
Right.
But that being said, it's a more expensive test, but it can be used on patients who are symptomatic as well as asymptomatic. So it's really good to use for surveillance purposes, right? The other test that's looking at active infection is the antigen-based test, and that's a rapid diagnostic test. So that's the one that kicks back a result in 10 to 20 minutes. So that's really good. It's a nasal swab. It's relatively inexpensive as well. So those are the positives on that side, but it's less accurate. It's about 20,000 times less accurate, actually. So you have to have a much higher viral load for that thing to pop positive. And for that reason, the indication for use on that test is a symptomatic patient within the first 5 to 7 days of onset of symptoms. And the reason why is because that's when your viral load is really peaking. And for that reason, it's not something that should be used for surveillance. So if you're an asymptomatic patient, and maybe you're concerned that you have, maybe you're concerned that you ran into somebody who had COVID and now you want to know if you have it too, that's probably not the test to use, right? It's not gonna give you an accurate result, and it could actually give you a false negative. And at that point, you're walking around spreading the disease, because one of the tricky things about COVID is you are contagious a couple days probably before you even start to show symptoms, and that's what's throwing everybody off with this thing.
Yeah, that's what I was gonna say. I always put myself on the line here, but I probably have had 5 tests, since March timeframe. Part of it is just not knowing, right? From that, and you think that you've got a fever or whatever, and some of the tests were early on, but the first one was a nasal phalangeal test, a PCR test, and I had to show symptoms, that I thought for symptoms to be able to get the test. The other one was like a mid-terminate, right, test, for a PCR test to be able to do it, or interior. And I guess those aren't as accurate because they don't get as much viral load on them as well, potentially, right? Because you're not going as far into the nasal
and
side.
I think, everything changed, right? Everything's changing and evolving. So initially we really felt like we had to go back there and tickle your brain to get the best specimen possible. Now we're pretty okay with just the mid-nares or mid-turbinate swab. Those seem to be performing really well.
And then I think I actually went into Sonora Quest Labs Bias, and I got a blood draw, right, to be actually get, a test done with it as well. And the difference between the blood draw is not a PCR test, right? A blood draw is, I don't know.
That's your third type of test. That's your antibody screen, right? So the antibody screen, generally, if you're testing for the IgG antibody, you're not looking for active infection, and you're really looking to see if you had an immune response because you may have had COVID, right? And that immune response potentially, we don't know for sure yet, but potentially could confer some type of immunity for future use. But that's a tricky subject, and maybe we can delve into that a little bit more. But that is your third test, is the antibody test. You've got the IgG antibody looking for past infection, IgM antibody, which might show present infection, but there's a lot of cross-reactivity with IgM antibodies, where it can pop positive, but it might be because you have flu antibodies that it's picking up instead.
So let's. I know we've learned a lot of lessons over the last year, from this. Have we progressed to the place where we have a rapid test now that is almost like a pregnancy test, where you actually can test somebody at home, right, to know if they, have COVID-19? First, do you test? Kathy, I'll take it to you first. Do you test when you're testing? Do you test? I've heard several ways, for antibodies first or antigens first, right, with this, versus a PCR test, right? You test for more if they've got antibodies versus whether they're positive. I mean, what is the known practice today, right, for knowing if you want to go safely back to work, from that perspective? How are they testing today?
Yeah, so it's a great question, Kent. And the good news is we have all those tests that Tom mentioned. The other good news is each test can be applied based on the purpose of the test. If you want to be diagnosed, meaning I have symptoms, your antigen or your PCR nucleic acid test would be the best tests. If you are trying to determine, I felt really unwell 2 months ago, I wonder if I had COVID, that's where the antibody test comes to play. There are some people who think, oh, I want to check my antibody response to see if the vaccine took or not. And that's what Tom was saying, it gets a little tricky. There are a lot of people who may have had COVID and never have detectable antibodies to SARS-CoV-2.
Mm.
But if you were to challenge them again with SARS-CoV-2, they would be able to mount an immune response, because there are two different kinds of immunity in our bodies. There's this adaptive immunity, and one part of it is antibodies, and another part of it is we call T-cell based immune response. It's a cellular immune response, and we don't have any good diagnostic tests commercially available to test for that. So Ken, if you or someone had COVID 3 months ago, their antibody was negative, but they had COVID, in fact, they were hospitalized. If you get the antibody test and it's negative, it doesn't mean that they're not protected. They still could be protected. It's just the cellular immune response that would be kicking in. And the same thing happens with the vaccine. I know of friends who have had 3 vaccinations for the hepatitis A virus and never amounted a detectable antibody. Does that mean the vaccine failed?
Right.
No, it just means that they don't have detectable antibodies, but if challenged, they would have the T-cell response. So each test has its own niche, and one size does not fit all. Yes, there are home-based tests that are like the pregnancy, little cards that are FDA, EUA authorized. One is a test called Ellume, E-L-L-U-M-E. They're in very short supply and very, very hard to get. And another one is this Binax Now antigen card. And there are ways that you can get that for home use. Some of them, they require prescription and supervision, telemedicine.
Right.
And then there's another home test that's actually molecular based. So PCR type, nucleic acid based, and that's Lucira Health.
Yeah. Tom, I'm gonna go back.
They
Sorry,
exist.
go ahead.
No.
Go ahead.
I'm just going to say they exist, but one size does not fit all.
Okay. Tom, I'm gonna go back to you really quickly. So in this time frame right now, and employers want to be assured that their employees are coming back to work safely, what is the test that you're recommending or doing on a regular basis as a protocol? I mean, I know that Christina and you and I talked months ago about passport cards, and potentially they would be green if you are safe and red if you were not safe, right, within a quarantine period, or you're positive from that. Are those type of programs being implemented out there today?
I don't hear about that very much, honestly. And I'm gonna hold back on this a little bit, because I was just on a webinar with the Arizona Commerce Authority, and they were talking about bringing people back to work safely. And it was Dr. Christ was on there, and she was, so I'll talk about what she said. She said it's a number of things, right? You're not just going to look at a test. You're really going to be focused on that quarantine period and bringing them back after the quarantine period. I'd love to be able to tell you that the RT-PCR test is the one to use. And it's a great test, and it's incredibly sensitive, but it is not a test of cure. It is a diagnostics test. And we have to be careful about that, because it does pick up very minute traces of that virus. So much so that you might be 20 days out from having had that virus or that infection and still be able to pick that up and pop positive. So we have to be careful. That being said, I'm not sure that the antigen-based test is the right one either. I think the best way to go is perhaps looking at the actual quarantine period and quarantining for that period of time, and then going back to work. Doctor, what do you think?
You're on mute, Kathy.
It's a really good point, Tom. I'll harken back to our prior new normal series that we had last year. And I said, you know, testing is often performed to mitigate our inability as humans to quarantine and our inability to really want to wear those masks and our real inability to want to physically distance. So we think we could test our way out of a lot of situations. But in fact, testing is one part of that tripod. And you're absolutely right. In that one part of the tripod, they all have their strengths and benefits. Some people say, well, antigen testing makes testing more accessible because it can be done right there and it's less expensive. So I'll do it 3 times a week on my employee workforce. Would that be okay? And there are different ways. It's a very statistical models, depends on disease prevalence in that community. If it's really, really high, maybe serial testing with antigens makes sense. Maybe RT-PCR, you swab them before they come into the building, and then they get their result the next day. Maybe that's the way if the prevalence is super, super low, because as you just said, the sensitivity of tests that are at labs such as Sonora Quest are really, really sensitive. They have great diagnostic performance. So it all depends. And I think opening up the workforce won't have one solution for everyone. School systems in certain communities will open up differently than other communities. Might depend on the variant strain, it might depend on disease prevalence, a whole host of factors.
Yeah.
The age of the kids, will they really wear masks?
Yeah, yeah. So there's other question I want to get into. We're going through this segment pretty quickly, but you know, Tom, I wanted to ask you about the variants in testing. You know, the current variants that are kind of going through, and Kathy, I know you're pretty versed on as many variants as we know about right now coming in to the United States. Are the testing systems set up so they can actually catch those variants right from that?
The real-time PCR tests are certainly, because we're looking at multiple targets with those tests. So it's not necessarily designed to pick up the spike proteins, which is what's evolving, what's mutating there. I think one of the interesting aspects of that, though, is that Thermo Fisher produces a platform called AttackMan, which looks at the spike protein, but also looks at a couple other targets. And it's interesting because that's how they're able to find these variants, is because it's missing on that spike protein target. It's not seeing anything there when it should, but the other targets are registering as being present, so you know you have a viral load. And because of that, they're saying, whoa, something's off here. And those are generally the specimens that get sent to the CDC for sequencing, and we're finding these variants, these variants through that.
Right. Kathy?
Yeah, thank you. I think hats off to laboratories like Sonora Quest and all the other major reference laboratories in our country, because they have stepped up and are assisting the public health laboratories by trying to identify these variants and then sending them on to the CDC. Or in fact, I know Quest and other labs have even contracted with our federal government to be able to do the sequencing themselves, which has been such an asset for us to be able to track the variants in all the communities in the United States. So it's heartwarming in knowing that we have a public commercial partnerships going on right now in the testing community, and it's going to help us better understand the diagnostic performance of the tests currently on market. The good news is the tests are performing well, and diagnostic performance has not suffered. So that's really reassuring.
Yeah, I think I did some early statistics, or looked at the statistics today, and it says out of the 28 million cases that we've had, there was 120 million that were negatives, PCR tests, and total test results was 350 million, 351 million, or 348 million tests in the US, which I think is, a good number, but it also signals that maybe we're still not testing potentially enough, right, from that as well, because that's just averaging 1 per person in the US, right, from that. But I mean, obviously, do we test children on an ongoing basis? We're testing more of a population that's probably more susceptible to the virus, right? From that, what are your thoughts, Tom, on obviously the number of tests that we performed to dates and whether it's underperforming or not?
Right. So a couple of thoughts on that. I mean, we have collection sites set up all across the state.
Yeah.
And I don't think at many of them that there's much of a line. I think you can get right in for most of these places now and get tested when you need to. If you have insurance, great. If you don't have insurance, that's okay. There's still no out of pocket as long as you either have symptoms or you feel like maybe you came across somebody who had COVID and you were exposed to it. Okay. No out of pocket. So that's not the rate limiting factor. But I think what we need to do is make it more accessible either in people's homes. And what we're trying to do now, at least for our school systems, is think about pooled testing, where we're pulling a bunch of these specimens from a bunch of different people and basically having them in one tube, testing that one tube and seeing if anybody in there is positive. And if they're not, great, everybody's clear. It really only works when you get down below 5% prevalence rates. It works really well if you get below 1% prevalence rates, which I think we're on their way to get there here pretty soon, hopefully. And that makes it really affordable at that point too. Right now, instead of it being a pretty expensive test, you're getting down and really approximating the cost or even dropping down below the cost of an antigen-based test. So
Right.
I'm looking forward to the days that we'll be doing that now, and we're talking to the state about that presently.
I mean, there's still an education issue a little bit because even on the vaccine, I want to go over the vaccine here in a minute, and testing as well. And maybe it's just my ignorance on this, but even when I get on Maricopa County's website and try to figure out even if we're eligible for vaccine and where to receive it, you hear about it's at Muni Stadium, you hear it's at Cardinal Stadium, but you don't know, am I eligible to get it? Am I in that class to get it yet? And also on testing, I know I can go to Sonoran Quest Lab and get testing. I know I can go, we've gone to CVS, right, and got testing as well, right, to be able to do that, to drive through. But it was kind of hit and miss for quite a while of like information of like where you get testing at from that. We knew that the Arizona sites would show up, for testing that's out there. But do you feel like there's better communication now, from the vaccine and the testing side, and people know where to go?
If you're asking me, I still feel like vaccine, I think Arizona Department of Health Services is doing everything they can to inform
Yeah.
people, but I don't know that that's getting out there at the level that it needs to be deployed. I think the Arizona Department of Health Services has done a wonderful job with their website showing people geographically where's the nearest testing facility to you.
Yeah. Right.
And thank God for people like Embry OBGYN, who's gone out there and really just opened up testing facilities all across the state and done a fantastic job of collecting those specimens. They stepped up. They stepped up. And without them, we would not be doing as well as we are right now.
So I want to move over to the vaccine really quickly. And has J&J been officially cleared as far as the vaccine, or is that tomorrow potentially?
It was submitted on the 4th of February, so the application was submitted.
And can you talk really quickly, Kathy, about, is the J&J vaccine going to be potentially better? I know it's different, right, than Moderna and Pfizer, just the difference between the vaccines.
Yeah, it's a really good point. Right now on market in the United States, we have Moderna and Pfizer BioNTech. Let's not forget the other part of that company that was responsible for that vaccine. And both of those vaccines use a messenger RNA technology where you actually have genetic material, the messenger RNA, in this vaccine that tells your body to produce the non-infectious spike protein. So basically that genetic material gives instructions to the body and says, hey, I'd like you to produce non-infectious spike protein. And then your immune system says, oh, okay, I see all this protein and it's foreign, so I'm going to produce an antibody to it. And that's how Moderna and Pfizer are, that's their technology. Johnson & Johnson uses a different technology. Instead of messenger RNA, they use a common virus that is a very common, harmless virus. And it's a vector, it's an adenovirus. So in that technology, they insert the instructions in this adenovirus vector, gets into your body, and once again, it gives instructions for the body to make protein of the S protein. And then the body sees so much of this protein being made, oh, it's foreign, let's produce antibodies against it.
Okay.
So those are the 3 vaccines.
Right.
The Moderna and Pfizer, as we know, were approved early on, and they have in the clinical trials great success rates. They prevent symptomatic infection, severe disease, and death in greater than 95% of the individuals who are vaccinated, which is really unprecedented when it comes to vaccines. Influenza never even comes close. Our influenza vaccine success rate is never that close. So it's just absolutely exciting news for the vaccine industry. But then you might ask me, well, that's what it's like in those really well-controlled clinical trials. What is it like in real life? Well, it just so happens that the New England Journal published an article yesterday or today from Israel. And Israel was one of the few countries that was able to vaccinate their population very, very fast and has probably the best coverage of a vaccinated population in the world. And they looked at 500,000 people who were vaccinated with the Pfizer-BioNTech and 500,000 people who were not. And they truly proved and confirmed that the effectiveness of the vaccine is exactly spot on what we saw with the clinical trials. So it prevented symptomatic infection, severe disease, and death. But a surrogate, a new proxy was in this study, they said, you know what? We also saw a decrease in asymptomatic infection, which then decreases transmission.
Right.
And the early clinical trials for Pfizer and Moderna were not intended to study the asymptomatics. So it's really exciting news for vaccines to say, hey, if you get vaccinated, first of all, get vaccinated. It's going to prevent you from dying, and it's going to prevent you from getting hospitalized. And I work on the front lines and I see patients with COVID. Please get vaccinated. It works.
Well,
Someone might say, oh, I won't get
good.
vaccinated though, because I still can get infected. Well, you still could get infected with influenza, but you still get vaccinated.
Right.
And you're protecting not just you, but your family. You reduce the amount of virus in you if you get vaccinated. There are so many pros of getting vaccinated. So that's Pfizer and Moderna. Johnson & Johnson is a little different. Its effectiveness is maybe in the 80s for severe disease and hospitalization. So not nearly as high, but still a great vaccine and much, much better than our normal influenza vaccine.
But it's one shot, and it's also stored at normal refrigeration,
yeah.
right?
Yeah,
as well.
exactly.
So potentially, I mean, at one point they thought that we could go down to the 1 shot, even if it was cleared for 2 shots.
Yeah.
Right. With the Moderna, do you think they ever relax the difference between the Moderna and the Pfizer to 1 shot?
I think as we get more data, we might be able to get there. Yes.
Yeah.
I mean, these vaccines are quite impressive. And I encourage folks, though, because I'm hearing such negative press about, well, the vaccine still doesn't change your life. Oh, it does. It does. Because you're not going to die from
You're
COVID.
not going to be on a ventilator. Right.
Correct. Yeah.
So the interesting thing, I kicked off a poll, right, with the studio audience, and I asked them, how many participants have had at least one COVID-19 vaccine, and 69% said they had at least one, right, which is great, right? And then I asked, if you were offered the vaccine today, would you take the injection? And 88% said they would take the injection, and 12% said they would not, right, from that. And how many have had PCR tests or rapid diagnostic tests that at least of zero tests? 19% said zero tests, 31% said one test, 13% said two tests, and three tests, 19%, and then five tests or more, we had 19%, right, from that. So, you know, it kind of, it's the gambit, but does it surprise you that people are still, I mean, I have a friend that's 77 years old. She has a very chronic disease, right, that's out there, kidney disease. And she's afraid the vaccine will affect her brittle kidneys, and she doesn't want to take the test. She doesn't want to take the vaccine, sorry, or from that. Is that a concern, right? I mean, obviously it's a concern.
There are certain patient populations that should not get the vaccine, but those are usually folks who have known allergies to the components of the vaccines, or individuals who are actively getting chemotherapy, not because the vaccine's going to harm their chemo treatment, it's because it won't really be effective because their immune system is suppressed from chemo. So we recommend wait until you're done with chemo, your immune system gets a little bit better, and then get vaccinated. So there are very few contraindications. Pregnant women can get vaccinated. So the 12% might be those folks that say, I'm going to wait to see if people turn into green Martians after a year. Well, that year is going to be up pretty soon because the clinical trial started in May and June of last year. So if you're waiting for the 12 months of follow-up, you're pretty darn close. So sign up for that vaccine.
I get it. So Tom, let's go back to you really quickly. So, you know, people are going to get, you know, the vaccine. The majority of people who need to get the vaccine are going to get the vaccine that's out there. How does testing, once you've got, you know, the vaccine, how does testing play a role, right, in that? Is, you know, is the vaccine only going to last a certain period of time? You know, I've heard that say that we may have to get a booster shot every year, right, for this, just like you do the flu shot, right, that's out there. How will testing play a role over the next year or two?
Well, to go back to what Dr. Petty said earlier, you know, the adaptive immune system is incredibly complex, and we can have people do a semi-quantitative IgG test. That's out there. That's something that's actually new. Before that, we were just doing a qualitative. Do you have these IgG antibodies? Yes or no. Now we're doing a semi-quantitative, which is kind of giving you a level or a range of yes, it's positive and it's strongly positive. But what we don't have with those is the CDC guidelines on what that exactly means. And if you have a strong reaction and you have a lot of the antibody, does that confer, at what level does that confer any level of immunity? So we're going to go beyond that. We're going to evolve beyond that. And so we're working with several companies right now that are actually looking at the adaptive immune response in different ways, looking at the T cells, looking at the types of IgG antibodies that are being produced, and trying to figure out who in this mad world is going to have immunity based on these inoculations, or just based on the infection that they have. Not only that, but by judging their immune response, at least initially, we can kind of place these patients into different cohorts that will help the doctors in the hospitals understand better who's potentially going to have a poor outcome to this and who might weather this storm pretty well. So those are things to come. I hope that we'll be developing those here in the near future. We're working with, again, as I said, a couple companies that are pretty hot on this right now, and we're providing them with some specimens so that they can validate their studies. And yeah, definitely more to come on that. It's really exciting. It's really exciting for me to be really on the forefront of the technology, and this is my opportunity in my life to really help our society get through a war. That's kind of how I feel about it.
Yeah, I get it. So one of the things I've been putting through my mind over the last probably couple of weeks or so, I do have, you know, a close relative that I'm around that has older that was diagnosed with COVID-19. Thank God he's come through it. But he's in a group facility around other people. And, we obviously want to visit him, right, on a regular basis. But, he's gone through a 2 and a 3 week quarantine period. He hasn't been vaccinated yet. That's one thing from this. And he's actually 84 years old. And I'm surprised he hasn't been vaccinated yet. But people that have been vaccinated, how long can they shed, right, from that? How long, do we need to be safe around people that haven't been vaccinated, people that have been vaccinated? What's the general rule of thumb? One thing I'm really concerned about is people are going to get vaccinated and say, hey, I can take the mask off, I can do anything I want, that's out there. And then the variants come in, and then our fourth wave comes in, right, as well. Does that concern you, Kathy?
Yeah, so let's start the vaccine, fortunately, does not have you replicate live virus. So unlike some of the vaccines that are out there where you actually do replicate live virus, the COVID-19 vaccine, you do not. So you never shed it. But in response to your question about if I'm vaccinated, can I go hop on a plane without a mask and not worry? It's complicated. So the first thing is, did you get 2 doses? The second thing is, did you wait 2 weeks after your second dose? Because that's when we've seen that you have your peak antibody response. And then thirdly, the world is not always about you. You want to also protect other people. So, and there is this low risk of being asymptomatic carrier, just like with influenza. So in order to be a good citizen, put on your mask and social distance, particularly for those people who are on the plane or in the restaurant who are not vaccinated. Right. I mean, we have a duty to our fellow humans around us to protect everybody. And right now, not everyone's vaccinated.
Get it. So really quickly, we're gonna have to wrap this up in just a few minutes, but in a layman's side, with the gentleman or the person I know that hasn't got the vaccine quite yet. I mean, obviously people can still bring it to them, even if the people are bringing it to him have got the vaccine, they could still carry it to him potentially, right? From that?
Yeah, very low risk, but yes. Yes.
Okay. Right. And part of the reason why they wear a mask, around him is because they don't want to potentially, with the low risk, transmit it to him as well.
Correct.
Right, from that.
Yeah.
Okay, I got you. And then really quickly on wrap up, let's just go to both, Tom and yourself as well. Tom, give me your prognosis of like going forward in the future, right? Over the next, let's just say the next 6 months or through the end of the year. Where do you think we are at from a testing perspective, and just from a testing perspective, where are we at, going forward?
Well, so judging on the history of this pandemic, as prevalence rates drops, our volumes drop for testing. And I was talking to Ken Komatsu, who's our state epidemiologist, this morning about that. And ADHS really wants to drive testing. They really want to make sure that we keep on top of this and keep volumes up there, because it's going to be critically important to really drive this thing into the dirt if we want to do that. And I think we all agree that we want to do that. So I'm optimistic that we can keep those testing levels up as we drive our prevalence rates down. I can tell you at Sonora Quest that we are modeling and we're following a lot of the national models. We're looking at everybody to try and figure out where we're going to go on this thing, but we're modeling a slight decrease. As I said, I think we're looking at about 12,000 is what our volume is right now, 12,000 a day, 10,000 a day, around there. We're anticipating getting it down to around 4,000 or 5,000 a day. But boy, I sure would like to see it stay up there so that we can make sure that people are know how important it is to get tested.
Get it. Great. And Kathy,
Tom, I completely agree, because also in order to open up schools, it depends on the prevalence rate in your community. Well, if you're not testing, you can't get an accurate measure of what is the test positivity rate in your community. So we really do need to continue to test in order to inform policy and opening up schools and other restaurants and what have you.
So I want to thank, both, I just want to thank everyone for attending the event this evening. And I want to take a minute to thank, Kathy Petty and Tom Leggett for participating in your great insights around COVID-19 testing, variants, and vaccines for the future. Did either one of you have any closing other comments that you wanted to say before we sign off tonight, from your perspective?
Yeah, I just want to make one comment. Variants, please do not be drawn in by the sensationalism of our media. Viruses will mutate. That's what they do. So please do not react and get anxious every time you hear a headline that there's another mutation. It's okay. We're following it. The vaccines are being adjusted if necessary. Testing companies are adjusting if necessary, but right now it's all okay. Variants will happen.
Great. And Tom, do you have any closing thoughts?
Really, I don't. I think this needs to be a multi-pronged approach. I mean, I'm the lab guy, right? So you would think I would just say, hey, get tested. But really, there's so many things that we need to do with social distancing, with contact tracing, masking, vaccinations, and testing to make this successful. So I'm hoping that we can all do that.
Well, I want to thank both of you for participating tonight and sharing your information. It was great. I'm sorry that we weren't able to get to all the questions tonight, but we will post the video on life365.health and on www.newnormal.buzz website. So please be safe and healthy. We'll be holding the New Normal webinar every 2 weeks. We'll be back with the next episode entitled Patient Engagement and Personalization on March 10th at 4 p.m. Mountain Standard Time. On March 17th, we'll be hosting a webinar with White Plains Hospital entitled Increasing Access to Care in Our Communities and Beyond. And on March 31st, backed by popular demand, we will be hosting Robert Jaron and Brian Scarpelli, providing an update on 2021 remote patient monitoring reimbursement codes, especially in light of COVID-19. So thank you very much to everybody and have a great evening.
Thank you,
Thanks,
Ken.
guys. Thank you.